Okay, it looks like we've reached critical mass, so let's blast off. I'm Richard Chamberlain, the chair of PharmSci, for those of you who don't know me, which is most of you. I wanted to welcome everybody to the 50th anniversary Homecoming celebration. And before I introduce the panel moderator, I wanted to put our celebration in context. In the 25th anniversary, I was in the chemistry department. I was introducing the first speaker of the year, and so I was touting the fact that we were 25 whole years old, and without thinking it through entirely about who I was introducing. So so I gave the 25 anniversary spiel and how far we've come and so forth and so on and so forth. And the speaker was Dudley Williams from Cambridge University. And so he got up and said, it's very impressive, but Cambridge was founded in 1209. So I believe in the Socratic method. Anybody know what the oldest continuously run university in the world is? It's not Cambridge or Oxford. I bet nobody knows. That's one of the oldest ones in this country, but that's a, baby compared to the oldest. University of Bologna in Italy. It was founded about 1,000 years ago. So imagine our descendants sitting here 1,000 years from now, having the 1,000th year celebration. Okay, that's enough for me. So I wanted to remind everybody that first and foremost, the bathrooms are right outside here. And after the panel, the panel discussion, we'll have a reception and there will be food. But we're not going to bring that in, I'm told, until this is over to make sure you all stay. Okay, so we picked a panel that's it's interesting composition. I just realized looking at them. We have the very first faculty member hired in PharmSci as one of them, and the others are our newest assistant professors. So we have the whole range in a sense. I'll leave it to you to decide which is which. Won't be as easy as you might imagine. I'm going to introduce Rob Spitale, who has been here since July, and he will moderate the panel discussion and introduce his new colleagues.
All right. So first, let me just say thank you very much for coming. We have an incredible group of scientists that are going to come up and share their scientific endeavors with you. But let me first start by saying that this is a really remarkable time to be in science. Today you're going to learn about bacteria, not only how we have good bacteria and bad bacteria, but also how we can reprogram viruses, which everyone knows are bad, to actually kill cancer. And further, we're going to hear about just how similar humans and fruit flies are genetically and how we can use that as a model system to understand human diseases. And finally, we'll end with understanding some childhood cancers. So first, let me start by introducing my first speaker in the panel, Professor Weiyan Zhao. The human microbiota is an aggregate of microorganisms. A microbiome resides in our human bodies. It's been indicated that bacteria outnumber human cells almost 10 to 1. Most of the microbes that are associated with human appear not to be harmful at all, but rather assist in maintaining processes necessary for a healthy body. However, as we are all aware, there are plenty of bacteria that can be harmful to our system, and thus, detecting and fighting them is a major challenge for medicine and research. This problem is especially important in the growing number of bacteria that have gained antibiotic resistance. Dr. Zhao is here to detail some of the roles that bacteria play in human physiology and pathology. Dr. Zhao completed his BS and MS degrees in chemistry at Shandong University, where he studied polymer surface and colloidal chemistry. Dr. Zhao then obtained his PhD in chemistry from McMaster University in 2008, and during 2008 to 2011, he was a Human Frontier Science Program postdoctoral fellow at Harvard Medical School, Brigham and Women's Hospital, and MIT. All at once? All those places all at once, or no? Okay. So, he joined the Department of Pharmaceutical Sciences in 2011, but since then, Dr. Zhao has been quite prolific, where he received numerous awards, including the MIT Technology Review TR35 Award, which is the world's top 35 innovators under the age of 35, and the NIH Director's New Innovator Award. So let me please welcome Dr. Zhao to the podium.
Okay, so I want to tell you something today that's going to freak you out. You kiss people, right? But you may not be aware of what that means to you. So what happens is that 80 million bacteria are exchanged during a 10-second kiss. As reported in a paper just a few months ago. To kiss or not to kiss, I think that's really the question now. So we still don't know much about what that means, but as I will show you, we think those bacteria do more good than bad. In fact, in your body, there is 100 trillion microorganisms, which is 10 times higher even than your human cells. Collectively, they would weigh like 2 to 6 pounds, which is much heavier than your brain. So they're just everywhere. So they're part of us and they're part of them. So they make up this microbiome, which is an ecosystem that plays an extremely important role in human health. So as I just mentioned, most of these microorganisms are actually very good. They help us to digest the food, they produce nutrients, vitamins, and they boost the immune systems, and they also regulate metabolisms. For example, they help to harvest energy and also to reduce fat in the body as well. And sometimes they even produce antibiotics to help us to fight with the real bad microorganisms. And that's why if this microbiome is misregulated or is not functional properly, for example, due to the antibiotics we take, you can actually have all sorts of diseases, including cancer, obesity, diabetes, and also neurological disorders. On the other hand, as we also know, there are numerous microorganisms can cause infection or disease. And here you just have a very long list. For example, the infection in the bloodstream causes hundreds of thousand people die every year here in our country. And that costs like more than $20 billion every year to our medical care. What's even worse is that some of these bugs have evolved and acquired this mechanism to get around of this antibiotic treatment. Okay? So in the future, the risk is that even a very simple infection that can kill us just because our antibiotics are not working anymore. Okay? Those are called superbugs. And it's real and it's happening. So it's being considered as one of the biggest threats to human beings. And it's also really a national priority. So as it turns out, we, human beings, are largely responsible to this antimicrobial resistance. It's just because the overuse or misuse of antibiotics. In the clinic in particular, as you're all very familiar, if you go to the hospital with a sort of like infection, right? So your doctor will give you some kind of antibiotics and see if it works. If it does not work, you go back, you get something else, right? Studies have shown this sort of empirical treatment, up to half the time, is inappropriate or it's just not right. So when it's not right, more people will die because they do not receive the right treatment right away. And those antibiotics are extremely expensive and it's very toxic because they also kill the good bacteria, as I just talked to you about. So there is a large ongoing initiative in the country whose goal is that in the future, the physician will not give you any antibiotics until they know you do have an infection and what kind of infection you actually have. So in my lab here at the Pharmaceutical Sciences Department at UCI, so we're developing very rapid bacterial tests that's going to allow the physician to diagnose and identify those infections very early so that we can make sure we give the patient the right treatment when they need it. Thank you so much.
So our next speaker is Professor Mahtab Jafari, and she's going to be talking about the genetic similarities between humans and fruit flies. When you think about it, humans and fruit flies are pretty different, right? But it turns out that nearly 60% of human genes are shared and studied are shared with fruit flies and humans, The reason is that fruit flies and humans use the same or similar genes to develop into adults. However, their short life cycle difference between humans and fruit flies makes them an ideal subject for study in the laboratory. Dr. Jafari's ultimate goal is to use fruit fly as a model system to add healthy years to human life. Dr. Jafari received her PharmD from UCSF School of Pharmacy in 1994 and completed a residency program in clinical pharmacy. In 2002, she joined Abbott Laboratories as a research scientist in the neuroscience department. At Abbott Neuroscience, she led studies on metabolic complications of the central nervous system. She joined Pharmaceutical Sciences at UCI in January of 2005 and developed a curriculum for pharmaceutical sciences. Her current research interest is anti-aging pharmacology, in which she uses fruit fly as a model system.
So what you see on this title is actually the wrong title. After Dr. Zhao's presentation, I think the title of my talk should be To Age or Not to Age. So just keep that in mind. But this is pretty much what I do in my lab. We work with botanical extracts and we are trying to find out if they can extend lifespan. So a little bit about our research. We refer to it as anti-aging pharmacology. Although in my lab we don't work with any pharmaceuticals or any pharmacological agents, we work with plant extracts. So we identify them, we try to find out if they extend the fruit fly lifespan, and I'm going to tell you why fruit flies are excellent model system, an excellent model system for this work. And then eventually we evaluate the mechanism of action and why they are doing what they are doing. And of course, with any basic science field, you want to make sure that it has a clinical application and humans can benefit. My goal is not to extend fly lifespan. I get this question all the time. I'm using them as a model system to learn about human aging. But how did I get here? Because as you heard, I was doing clinical stuff. So until really 2005, I was a clinical researcher and I was working. I hope I'm pushing the button. Oh no, I'm not. Oops, I do it. Okay. So I was working in cardiovascular diseases, neurological diseases. I never did cancer, maybe a little bit of work with diabetes. And then I finally figured out, after 15 years of doing that, that all these diseases of aging fall under the big umbrella of aging. So what if rather than focusing on one disease and another disease, focus on aging and trying to slow the aging process? And this, in a nutshell, is what we are trying to do in my lab. And in order to extend the lifespan, you may want to, the first question that comes to your mind is that, okay, this is a great idea, but this is almost like science fiction because we don't have any examples of exceptional longevities or, really long lifespans in the nature. And the answer to the question, the comment, I found a pointer here. I found a pointer, but I don't know how to use it. Oh, here we go. So starting with the fruit fly, which is the model system that I work with, the longest life is 6 months. And you can go down, humans, 122 years. This is the longest lived documented human being. This is the cute Madame Jean Calment, who passed away a few years ago. And then we get into the immortal jellyfish, which is biologically immortal. So we do have examples of immortality and exceptional longevity in the nature. So why not in humans? So this is what we are trying to address in my lab. So as we age, so this is in our 20s, 40s, and 60s or 70s. So as you can see, I'm not going to go into any details of what is happening. What we see here is aging. And if we are lucky, we make it over 120, and we look like Jean Calment, who passed away a few years ago. And she was quite healthy, actually. I'm not going to talk about her lifestyles because I have some students in this room. She was a smoker. So let's not focus on that. So the goal of my research is not to get to 100 and look like this. This is my goal. and I'm not just talking about looking like her. You know, she's gorgeous. Not looking like her, but maintaining the function and the youthfulness of a young body. So this is what we are trying to do. So now I'm going to get to the did you know part. So Rob already gave half of my talk, so I'm going to go over the next few slides very, very fast. Thank you very much for doing that. But the question that I'm going to ask you is this. Did you know that we share about 75% of our disease genes with fruit flies? Did you know that we have models of Alzheimer's, Parkinson's, schizophrenia, diabetes, addiction in fruit flies? Meaning that fruit flies can have these diseases and we can study them to test drugs that are used for these diseases. And I'm not going to bore you with this slide, with this diagram, which was adapted from Science in 2010. So this is just to show you how similar we are to fruit flies. So this is us, mammals, these are flies. This is the dietary restriction pathway. And if, without going into details, just pay attention to the names and numbers, and you will see that they are very similar than us. But fruit flies don't share just longevity pathways, because this is the focus of my lab, that they share some common aging pathways with humans. Fruit flies, or us I should say, humans behave just like fruit flies. And let me give you just a couple of examples. So how many of you guys in this room have been rejected, male or female, and got drunk? Nobody did. So this is exactly what a fruit fly would do. So fruit flies, when they get rejected, and they have the choice of drinking or food, and these are hungry fruit flies. So you would think that they go for the food because they're hungry, but no, they get drunk. So their behavior is very, very similar. Again, this is alcohol preference in fruit flies, and they just go for the drink. They have no desire to eat. They were rejected. They go for the drink. We did this experiment in my lab. Please don't ask me why. I don't know. I had a very outside-the-box student 2 years ago who wanted to do that. So what we did is that younger male fruit flies were mated with younger females. So the male flies live slightly longer when they mated with younger females. I think this explains why you see so many older men out there with much younger women. But the news that I have for them is only slighter. So don't go crazy. The opposite, female flies live considerably shorter when mated to younger males. Anybody knows why? You're taking care of them? No?
Can't keep up with the guy.
Can't keep up with the guy. No, these guys stress them. So female flies live considerably shorter when they date or go out with younger fruit flies. We haven't published this work really. Because I'm trying to find a very good journal to publish the work, and we also need to do some mechanistic studies, because I can't just publish this. But that explains some behaviors in our community, and I'm not going to say where we live, but I think we all know. So this is why I chose fruit flies as my model system. And without again going into any details, this is what we have developed over the past, I think this is still work in progress. So since 2005, We have been screening, trying to identify botanical extracts that will increase longevity, and this is the algorithm that we developed, and the algorithm is very validated because other people in other labs and other countries use our algorithm to identify botanical extracts and pharmaceuticals that might extend lifespan. But after 10 years of doing this is where we are. So we have identified 4 extracts that can increase the lifespan, some of them at the expense of something which I don't have time to explain, but I will show you a few slides to see what I mean by extending the lifespan at the expense of something. But these extracts are Rhodiola, rosea, rosea, curcumin, which is the active compound in turmeric, and green tea. And I don't have cinnamon here, but cinnamon is also another extract that we worked, and our paper was just published 2 weeks ago. So I'm going to show you in the next 3 slides just snapshots of our work. So this is our work with Rhodiola rosea. We have a few papers suggesting that Rhodiola rosea extends lifespan, and it has been a consistent increase in lifespan. So when we first published our paper, our first paper in 2007, other people kind of repeated our work in other model systems, worms, yeast, worm yeast, and snails. And they also observe lifespan extension with Rhodiola. And Rhodiola does that without any health expense, meaning that our fruit flies are healthy. I think they look happy, right? I have some students, they look pretty happy. And they just live a very good life. And we have given Rhodiola to all the fruit flies because this is a critique I get that, wait a minute, you start giving Rhodiola to kids, a bunch of fruit fly kids. And I said, no, I've given Rhodiola to all the fruit flies, and we've been able to slow the aging process in them too. Another, as I mentioned, turmeric is another compound or curcumin that we have worked with. And this study was, to me, was very interesting because it was a parallel study with some colleagues in Korea who did the same study in a different lab environment, different strains of fruit flies. And what we discovered was that the lifespan extension with curcumin was sex-specific. In my lab, we were able to extend the lifespan of male flies not females, and in KJ, my collaborator lab, opposite result in Canton S flies, meaning that turmeric extended lifespan, but only in female flies. And then this is our last work that, as I said, just got published with cinnamon, showing that cinnamon can extend lifespan. But what I thought it was very interesting with cinnamon was not just the lifespan extension property, but also the fact that cinnamon improved physical activity. And it kind of answered one of the questions that, or addressed a comment or a question that we always had about cinnamon and we didn't know why. How many of you know that if you're in a cold temperature, you can take cinnamon and it keeps your body warm? So some people from traditional medical practices in China, in Iran, that's where I'm from, they definitely use cinnamon to stay warm in harsh and cold temperatures. And we showed that the reason for that is because cinnamon increases heat shock protein expression. And this was, I believe, to date, has never been documented. And finally, green tea. So how many of you drink green tea? Okay, so if you are a woman, no problem. Drink as much green tea as you want. But if you are a man, drink green tea if you don't want to have kids. Because what we showed, or you're done with having kids, no need for more kids. Because what we showed was that green tea extended the lifespan of male fruit flies. And this was very funny observation that they extended the lifespan of male fruit flies only when they are with female flies, because we separated them. So when male flies are alone, I think this goes back to the stress a little bit, they get no benefit from green tea. But what we also showed, this was the work of my graduate student, is that the green tea extends the lifespan of male fruit flies, but it decreases their reproduction. So we have looked at various pathways in reproduction. We've been counting sperms. We've looked at offspring. So green tea is not really a good idea if you want to have kids, or if you're a fruit fly, I should also say that. So I would like to end my presentation by acknowledging, all my students, and there are some of them are here. So Dr. Scheiner, I say former project scientist, but because he's no longer in my lab, but we're still collaborating. Terry Lopez is my graduate student, my newest addition to the lab, Dr. Pascal, who's not here, and of course all my students, because working with fruit flies is very, very time-consuming and labor-intensive. And if you don't believe me, you can ask my students. Anyway, so since you have questions at the end, I'm going to stop right here. Okay, if you thought this was funny, did you think this is funny? Yes. Okay, read this one.
Our next speaker is Professor Yongjik Kwon, and he's going to be talking about synthetically tamed viruses for cancer therapy. We all know how deadly viruses can be. They have the capacity to make us incredibly sick and even induce death. But what if we could tame that strength of the virus and use it for good? Dr. Kwon is going to describe his efforts to re-engineer viruses and turn them into cancer hunters. Dr. Kwon received his PhD from the University of Southern California in 2003. He was a postdoctoral scholar at UC Berkeley from 2003 to 2005. He set up his own lab at Case Western Reserve University and was there until 2007 when he moved to UCI. Since coming to UCI, he has won the Faculty Early Career Development Award from the National Science Foundation and the Medical Research Award Gabriel's Angel Foundation for Cancer Research. He also runs the Biotherapeutics and Engineering Laboratory BioTel, which seeks out novel methods to develop smart therapies and diagnostics.
All right, thank you, Rob. So today I'm going to talk about a little bit exciting science that has been already known to public. So here are the examples. So I'm pretty sure many of you have watched this movie. But what I want to really say is that Hollywood always loves the crazy scientists and conspiracy theory, things like that. But we are really trying to make really emerging therapy to help people. Okay, and one of the forms of emerging therapies in the clinic even is gene therapy. And to be surprised a little bit, there are about 2,000 clinical trials going on already around the world. And surprisingly, many of the clinical trials are using viruses, including adenovirus, which is a common cold virus. Everybody in this room may see them all the time every year, and the retrovirus, and the vaccinia virus. That's the very first virus used in the vaccination against the smallpox. And also adeno-associated virus, and the pox, and the lentivirus, even herpes. Okay? And even in the late 2012, the European Union approved the first virus-based genetic drugs to be sold in the market and used in the clinic to cure lipoprotein lipase deficiency. Okay? And most clinical trials are the targeting cancer because that's a really big deal. And obviously, the traditional therapy did not work out very well. And the monogenic disease such as cystic fibrosis, and infectious disease such as AIDS and things like that, cardiovascular disease and so forth. So how the viruses can cure cancer? So one of the ways the virus can cure cancer is basically to induce the death of the cells directly or by activating sensitivity toward certain drugs. So that when you give the drug, that drug never works on the healthy cells, but the cancer cells got infected by viruses and the virus activates the drugs to kill the cells. Okay? Another way is called oncolytic viruses. These viruses are engineered to be proliferated only in cancer cells. They just infect the cancer cells, and they just proliferate crazy, and they disrupt the cancer cells and spread to the next cancer cells. Another way to cure the cancer cells by viruses is to boost up the immune systems. One of the ways is to engineer the T cells by putting the viruses to recognize particular cancer cells, so that usually T cells can never see the cancer cells, that's why the cancer cells survive, but we can re-engineer T cells by putting some viruses to re-engineer the cellular properties. So basically, when you are saying we are healthy, doesn't necessarily mean that everything is the same. Actually, a lot of things are happening in the equilibrium. So basically, in our body, there will be very well balanced that's between survival. So every day we get the new cells, but also we are losing a lot of cells because they are too old. But in cancer, basically, we don't have this balance anymore. Basically, there are a lot more cells to survive rather than just disappear in balance. So there's a lot of cancer therapy trying to enforce the cell loss by putting a lot of drugs, using chemotherapy, or putting some rays to cure or kill the cells. But the problem with this conventional therapy is that we have to put a lot of forces toward death. That's quite difficult to achieve, and often we need to put a lot of drugs, and there will be a lot of side effects, and we are already suffering from the resistance against the drugs. So one of the cancers that I want to talk is the Philadelphia chromosome. That is very well known as one of the major causes of many forms of leukemia, including chronic myelogenous leukemia and some other subsets of leukemia. So there is a very good drug for this form of leukemia that's called Rebalancing or imatinib. Okay, that's basically very rationally designed chemical drugs. So this drug has been working great, and a lot of people are really excited about this drug. Okay, but one of the problems with this the drug is that this drug cannot kill leukemia cells. It somehow stops growing the cells so that this drug basically keeps all the cancer cells. And also, meantime, the cancer cells are smart enough to develop resistance. So that has been a huge problem. So my laboratory at UCI in the Pharmaceutical Sciences here, what we are really trying to do is not only just pushing toward the task, but we have to relieve a little bit on the survival process. So basically, we want to express some of the DAS proteins in the cells. At the same time, we want to silence the oncogenic activity. By doing this, we can get the really physical therapy by putting really small amount of drops, not necessarily putting loud materials at the same time. And because we are tackling two multiple pathways so that the cancer cells can really develop the resistance at the same time toward the multiple pathways involved. So what we are really trying to make is a magic pill, right? By putting the viral particles as template, and then we put some synthetic materials, so that we call this a chimeric nanoparticles. So we use the adenosis virus, which is one of the pathogenic human viruses, but there has been no documented disease caused by these viruses. We just use this AAV to express the DAS proteins, and then we put the secondary materials which reduce the oncogenic activity. So as a result, we had polymer and the virus chimeric materials expressing DAS proteins at the same time, reducing the oncogenic activity. So basically, this starts getting in, and then just express some suicidal genes and silence the oncogenic activity. Basically, that's what we really designed. And the really interesting aspect of this approach is that these materials are so specific because these materials will be efficient when there's a cancerous activity going. So in this case, when you put our materials in the non-specific cancer cells, there's no effect. But when we look at the cells incubated with our materials, with the Philadelphia chromosome positive leukemia, we can see that there was really significant impact on these cells, basically killing the cells with the cells having this particular form of chromosome mutation. So we put these materials in the animal, and as you can see here, no treatment, and that's protein only, and oncogenic activity silencing only, and our nanomaterials, as you can see that, our animals were doing a lot better, and extended their life actually much more significantly, and almost like stop the growth of the cancer cells, basically. Then when here we talk about they use these materials for human patient, and maybe people will raise the question, hey, this material is safe. Nobody want to get injected by the viruses, of course. So one of the big provocation is immune responses, because I'm pretty sure that most of the people in this room will have the immunity against the viruses, right? So we tested these materials, by putting these materials into the animals, and we collected the serum. And then we tested whether this animal ever developed immunity against these viruses. The result is quite surprising and promising. So our materials did not get recognized by the immune system, and also even did not provoke immune system at all, upon even the repeated administration, which is quite safe, and we believe that this is a really important aspect to move on to the clinical trials. So what I want to really say is that viruses in nature make us really sick, like recent Ebola outbreak. But if we just combine our scientific tools to make really well-engineered and designed materials, we probably can use these deadly viruses to save people from the really deadly disease such as cancer. Okay? Thank you very much.
Okay, so now we're going to move on to our last speaker, and then we'll have the panel of speakers come up here and they can answer all questions. So the next speaker is Professor Claudia Benavente. She's going to talk about childhood cancer or retinoblastoma. Sight, more than any of our senses, helps us navigate the world around us. In a single glance, lasting a fraction of a second, our eyes work with our brains to tell us the size, shape, color, and texture of an object. Dr. Benavente focuses on a form of cancer that attacks the eyes of children called retinoblastoma. And she's going to tell us about her desire to understand the epigenetic causes of this type of cancer. Dr. Benavente received her PhD from the University of Arizona, specializing in cancer biology. She then went to perform postdoctoral studies at St. Jude Children's Research Hospital, where she focused on childhood cancers, including retinoblastoma, like she'll speak of today. She was a Fulbright Scholar and the recipient of the coveted NIH Pathway to Independence Award. She joined the PharmSci department in 2004. Her laboratory is interested in understanding the molecular and cellular mechanisms that drive tumor progression.
2014.
Sorry, 2014. Sorry, excuse me. You haven't been here for 10 years yet.
Yeah.
And I'm actually not going to talk much about retinoblastoma, but I'll talk about why it's a good idea to study periodic cancers, even though they're not very common. But all of you are probably familiar with cancer. You've all heard about cancer, and you probably know people that have been affected. But do you know just how big of a public health problem cancer is? If you're a woman, you have 1 in 3 chances of developing cancer sometime throughout your lifetime. You're actually 1 in 5 chances of dying of cancer. If you're a guy, you actually have much more chances of getting cancer. You're 1 in 2 and 1 in 4 of dying of the disease. So this is a huge health problem in this country and worldwide. It's second only to cardiovascular disease, and they're always fighting to that first place, which is not a good thing to do. Many of you have also heard the terminology of when someone has cancer, they're fighting against cancer. And did you know that this actually came about back in 1971 when Nixon signed the National Cancer Act? And this was all termed like the war on cancer. So now the scientists were all at war with cancer. But piggybacking on what Dr. Pond just spoke about, do you know that cancer is actually yourself? Cancer cells are actually your own body cells going rogue. They have blocked the ability to die when they're supposed to die, and they just keep on dividing like crazy. And so this thought of you're going to go fight cancer, you're actually at war with yourself. So how about let's just restore cellular balance? Let's restore that harmony that Dr. Pond was talking about, balancing death and survival together. Now, did you know that even though retinoblastoma, pediatric cancer of the eye, only affects 20,000 kids worldwide every year, it was through the study of this very rare type of cancer that one of the most important genes in cancer development was discovered, and that's the retinoblastoma susceptibility gene. See, we're quite clever. We don't like to remember a lot of things, so the retinoblastoma is caused by a gene called retinoblastoma that it encodes for a protein called retinoblastoma. So you've learned a whole lot of cancer biology right now. Just know retinoblastoma, it's very important in cancer. And as a matter of fact, this protein regulates a pathway that is deregulated in virtually every cancer, not just childhood cancers, but also adults. And this is what a kid with retinoblastoma looks like. You can actually visually see the tumor in the eye. And this is the pathway that regulates. It regulates cell cycle, so cell division. But for many, many, many years, we thought that most cancers are actually a genetic disease. And you know, it was only in 2012 that the group that I belonged discovered that actually cancer is much more than just a genetic disease. And it was through the study of this very rare type of cancer that we found that cancer is also driven by epigenetics. And I'm going to take just a little bit of time explaining to you what epigenetics is, because you're going to start hearing it a lot in the news, because everyone 10 years ago started knowing about DNA and whole genome sequencing and how genetics is very, very important, but epigenetics is just as important. So think about this. When you're conceived, you come from one cell, and all of your DNA is now replicated, so copied exactly the same throughout all your cells. So you have all the same genetic information in every single cell of your body. How does the lung know that it's lung and it's not skin? How does your neurons are behaving like neurons and not like your pancreas? you're not having insulin secreted from your neurons. That will be pretty bad. All of that regulation is actually happening through epigenetics. And it was, I just want to refer to the study of retinoblastoma that we were able to prove for the first time that epigenetics can be a main principal driver of tumorigenesis. All thanks to this periodic cancer. And so this is part of all these things that are highlighted down here is how you differentiate. Like this is all the epigenetic machinery that is being differentially expressed, and now I know it's like getting into big scientific terms. But those are the difference between brain, heart, muscle, and the thymocyte. Is like all the epigenetic machinery that's being expressed. And so through that, it actually allows us to have a different tool, a different target through which to try to treat and restore the balance in cancerous cells. Also, and I didn't start with this, but in general, cancer is a very common disease. But the vast majority of the people that are affected by cancer are adults, and the mean age of diagnosis is 67. So you're fairly well seasoned when you're likely to get cancer. And because the vast majority of people that get cancer are adults, the vast majority of the force in going in to try to treat cancer goes into studying adult cancers. In fact, only 4% of all the U.S. funding goes to try to come up with therapeutic approaches for childhood diseases. There's 60% of all the money that goes into cancer research and trying to find out cures comes from pharmaceutical companies. There's not a single pharmaceutical company in this country that does cancer research. A lot of times, even when we try to go and ask for drugs so we can test it on our periodic patients, they'll say no, because it's too much of a public relations issue if anything goes wrong. So I do want to take the time to bring a little bit of awareness of where this comes from. But the importance, I mean, you could have said, well, there's just a handful of kids that get cancer every year, so why would it be important? Maybe it is more important for us to be studying adult cancers. But I just show you that through the study of this cancer that is very, very rare, we've actually come up with great discoveries that don't only affect the retinoblastoma cancers that we're studying, but that are actually extrapolate to cancer as a whole. Retinoblastoma susceptibility gene actually affects every single cancer known today. And you saw that first little newspaper article where it says that when the bill was first passed, it was put into $1.6 billion to cancer research. The funding today for cancer research is $4.9 billion. That's 3 times what it was back in the day, but it has stayed the same for the past 10 years. So how you can help us move forward for cancer research is, contact your people, your congressmen, your senators. I'm making a little bit of a political speech here, but say, you care about cancer, this is how you do it. Like, put money towards research. And not put it down. We can do some. Okay, and thank you.
So now I'm going to invite our panel speakers to come up here and sit and subject themselves to the firing squad. So if you have questions, I want to encourage you to please feel free to raise your hand. Let's keep order. We are in a classroom. But yeah, if you have any questions for any of the panel, feel free. Yes? Fire away.
For Professor Jafari.
Yes.
Why do organisms die? It's not necessarily, there would be ways of replacing cells like the jellyfish. So there's an evolutionary advantage of some sort to cycling through, giving the next generation the chance to consume the resources. But what causes the great variations in lifespans?
I knew there would be many questions that I don't have an answer, but it depends on the question of why organisms die. Like fruit flies die, we don't know why they die. So I'm working with an organism in my lab, and we don't know why they die. So we look at cancer, we look at many other reasons, we don't know why they die. But the idea is the maintenance. So I think if I'm working with botanical extracts, but for instance, botanical extracts are not the only solution, or they don't provide the only solution. So in order to not to age, you have to maintain the function. Cardiovascular disease is the number one killer in the U.S. So how do you prevent that? The future will be in stem cell research, because you are going to you're going to replace the non-healthy cells with healthy cells. But I don't really know the answer to your question, a very precise answer. We just know that as we age, the genes in our body that were dormant as we were young, because we had the power to fight them, to fight those diseases, they become more robust. And we get cancer, we get heart disease, and we die. But we shouldn't die. We shouldn't age.
Yes?
We have to age somewhat, right? I mean, in order to go from an infant to an adult. So what's the optimum age that you want to stop aging?
I recently gave a talk, and the title of my talk was "How to Live to be 200," and I had a full room. I mean, the people were standing in the back, and I said, I was just kidding. I mean, I was just trying to get people in this room. I don't know what the optimal age is, but I would say that some of the babies that are born right now, and this has been proven, they're going to live up to 150. And I would say maybe 10 years from now, we would say up to 200. And this is not just because of the advances in medicine, that explains part of it. But I think, again, aging and not aging is multifactorial.
What about like 25?
I'm over 35.
Yeah.
25, people who are 25 now.
Yeah, just want to, that's
We'll
it.
just stay 25.
Say 25.
Yeah, 25.
What's wrong with that?
There are some really good examples in some Hollywood movies, but not in basic science lab.
Guess we don't know what the perfect age is to just shut it down.
I think the perfect age is the age that your organs can still function, and they are maintaining the function, the youthful function.
25?
35.
30. We'll make it 30. Okay?
That's the end. That's the time to get that vast knowledge.
Yes, please.
Yeah, actually, a question for Dr. Kwan. It seems like an emerging field of cancer therapy is negating the immunosuppressive effect of the cancer cell. For example, antibodies specifically targeted to that portion of the cancer cell that is immunosuppressive, the antibody neutralizes that. So the question is, there been any thought of combining the viral treatment with other agents that counter the immunosuppressive effect?
Well, that's a really good question. So what I believe as a scientist and also the researcher working on the cancer therapy problem is that we may have a lot of tools already. We may know a lot of things about the different branches of biology and our body. But we've been kind of working on just one modality, one tool probably, like immunotherapy, chemotherapy, radiotherapy. But my understanding is that the cancer is so diverse. So there shouldn't be any kind of just one drug taking care of all the problems. So what I really believe is that we have to somehow combine a lot of different therapeutic modalities and then kind of working together in a very synchronized and harmonized ways. So one of the examples I showed you today is kind of manipulating multiple genetic events at the different times and different levels. But we do working on the combination of chemotherapy and gene therapy, and combination of immunotherapy and chemotherapy, even working the two different kinds of drugs, not really known to work each other, but we want to just combine together, normalize, so that we can really tackle the cancer cells at the different levels and together. So I think what you really raise is a really important question that I think a lot of people are really understanding and appreciate that point.
So I have a follow-up to that. So you talked about certain viruses that will replicate in cancer cells, not in normal cells, making them lytic to cancer cells. What are some of the hallmarks of cancer cells that would make those viruses be selective for those cancer cells versus normal cells?
Okay, so basically, when cells become cancer, there are a lot of things happening. And one of the things happening is they really change the profiles of the sequence of molecules that really modulate cellular interactions and communications. Those kind of molecules we call the cytokines. And some cancer cells actually kind of doing a little bit different ways to the general-induced materials. So when we design the viruses that actually can survive or cannot survive in the presence or absence of cytokines, you can make these viruses even much more surviving, a lot more proliferating, a lot more spreading. So that's how the people design the materials in general.
Yes.
I'm not a science student, so this is really general. But I read something in a paper recently that someone did a study somewhere recently where they dug up some soil and somehow they managed to separate all these, I guess they were bacteria, and isolate them and grow them. And supposedly they think they found several different bacteria that can attack these resistant bacteria that we have today.
Yeah.
Yep.
Yeah. Well, it's very impressive that you follow up this scientific articles. Yeah, it's one of the most recent, events. So one of the challenge for antibiotic resistance is because the drug library we have right now has been exhausted, right? So when we isolate a new compound, you do screening or somebody else do screening, you always get exactly the same drug that the bacteria has already evolved mechanism to get around that. So people are trying to go to other places, they go to the forest, they go to like deep sea, and in that paper, for example, they just discover some of the new bacteria they can produce this antimicrobial agent, that's gonna get around this resistance. So yeah.
Does that look promising then?
The activity isn't a coincidence. Bacteria and other organisms produce toxins that kill other bacteria that they're competing with.
Exactly.
And so that's been a strategy for a long time, a successful one until fairly recently. And pharmaceutical companies have largely dropped their antibiotic discovery programs because there isn't enough money in it. But that's a tried and true way of finding antibiotics, looking for bugs that make antibiotics to increase their selective advantage in their little ecosystems.
So Leon, to take that a step further, let's say that I give a bug a drug and it's not resistant to it. What's the prediction for how long it takes for a bug to become resistant to a drug?
I don't know. So I'm not a evolution researcher, but I guess it really depends. Depending on how hard you push.
The reason I ask that is because in the article she's referring to, it said something on the order of like 50 years.
Yeah.
Which seems like a long time, but
Right.
I don't know if that's real or.
You can use fruit flies and do it much faster.
Yeah. But one thing we do in the lab, we call this like in vitro evolution. Rob and I are working on lots of this where, for example, you want to isolate something, so you want to develop a new gene or new function. You start with bacteria, and you can really treat with a drug or something else that you can really push to guide their revolution to particular pathway, so to develop new functions. Maybe that's kind of potential approach to develop new agent for antimicrobial resistant pathogens.
There's a lot of research that's being done now where people are doing just like Professor Zhao said, where they take a bug, they give it a drug, and then they find out who survives, and then they isolate all those that survive, and they say, what's different about them? So now we can predict how the bacteria are going to respond to the drugs that we're giving them.
Yeah. However, so I want to mention that, so our government these days is very conservative to study this, so-called gain function. So nature does not have this function, right? But you want to do research to make something to have some new function, it can be very dangerous sometimes, right? So we had this, found flu virus a few years ago, right? It can be very deadly, right? So people are very careful with this, sort of thing.
Come on, I know you got more questions. Nobody gets off this easy.
Would it be appropriate to ask about the drug companies today? Like, you read all these articles and I have no connection to any of this to know how true what you read is, where the drug companies are so driven by profits like all the other big corporations that they're no longer making a lot of the drugs or researching drugs that used to be so common. You know, they have, what is it, the hepatitis drug that's like $1,000 a pill or something. And is the government doing anything to try to counteract that? Or is it totally drug companies?
That's why, whenever I hear somebody say this, that's why I say you should give all your money to academics, because
Can I respond to that?
Yeah.
So,
We were just talking, you said.
yeah, the drug companies are in business to make money. And, you know, I'd advocate some kind of nonprofit. I mean, they actually, a CEO of a drug company is breaking the law if he or she makes a decision, a financial decision that will hurt the bottom line of the company. That's you know, the rule. So I've in my career I've trained probably almost 50 PhDs who have gone into industry and become medicinal chemists in big pharma or biotech. And most of these people genuinely want to, you know, help by developing useful drugs and things like that. But if a drug, either a specific drug or a class of drugs or even a target like antibiotics isn't going to make enough money and they can make more money somewhere else, they sort of are obliged to go off in that direction. So I think it's a problem with the basic structure of the drug discovery mechanisms in this country is that, and I don't blame the pharmaceutical companies. They're not any worse than any other big companies, but I think that's not a field that ought to be so profit driven.
So do the insurance companies have a lot of say on a lot of that too, based on what they're willing to spend money on as far as what people are covered?
Well, I think that also depends on whether they're a for-profit thing or like a Kaiser, who does have to watch their dollars, but also.
I'm just going to add something. With Affordable Care Act and the reality that we are going to have more millions and millions of people in this country who are hopefully insured by now, and we keep talking about the insurance for these people, but what is going to happen? Now that I have insurance, I didn't have insurance, now I have insurance, I can go see a doctor. And if I have diabetes and hypertension and a little bit of a headache, my doctor is going to give me a prescription for pharmaceuticals to treat those diseases. So who is going to benefit? Pharmaceutical companies. So my hope is that now that they're going to, I don't want to quote any number, but we are talking about additional millions of dollars in profit for pharmaceutical companies. So my hope is that they dedicate some of that money, some of the profit to new drug applications, not making another Me Too drug, another hypertension, another same class diabetes. And FDA mandates that they do research in orphan drugs, which are like the neuroblastoma. I mean, they are not that profitable, but we don't have enough of that. And I think that a duty of Affordable Care Act is also to really look into what they do with that profit. And hopefully we are moving towards a better R&D in pharma.
I'm wondering what the next step might be for mobilizing society, because when Nixon proclaimed the war on cancer, I read Edward's Malady about cancer development, and basically there were lots of scientists that got together and created a message that mobilized people to do something. They created NIH, they really got together and said, let's do something about this. So what do you think, and you ask the various panelists, would be the best way for us citizens to band together to start a movement, to get the word out there? Because I think that's what it takes. It starts, local, and it just gets bigger and bigger. I think that's what the Susan G. Komen, activities have done. So many people have participated in that to raise funds. And if you have any thoughts, I'd love to hear about them, and just how can we all get mobilized?
So
Yeah, I'm looking at
Just go down
Well, in my opinion, I think public awareness is really important in some senses because when I talk to a lot of people, I mean, either they are not really interested in or they don't have any background about science, so simply they just believe, I mean, they just regard all these things like, you know, not your own word. So they just say, okay, this is science. Okay, I'm not scientist, things. But, I mean, as I just, you know, they mentioned on the very first part of my talk, I mean, there are a lot of movies that people really understand. That's not really completely fiction. It's based on something happening there. I'm pretty sure everybody with a little bit of education should be able to understand things. That they really have to think that what really happens in pharma or the research, that's a part of their world. And that they really have to start and understand. And whenever there's a chance, like you mentioned, you know, human activity, just getting there and getting to know the people. I think that's a really kind of important thing. And it does not require, you know, in my opinion, tons of money, tons of time. Just getting involved there and just talk to the people and then bring up the issues. And if there's a really big deal, that they have something kind of practical agreed upon, and then talk to the kind of assembly members and hear what you believe that you should do something about it. I think that's something that we have to do. I mean, we know all these steps, but the problem is that we actually don't do that actually.
I agree with Young. I think education is the key, and we don't do enough of that. But we have good examples. I recently read an interview by actually a dear friend of mine, Dr. Sabeti at Harvard, who just sequenced Ebola. And they said, when did you start learning about Ebola? He said the movie, The Outbreak. So that's where I got my first education about Ebola. And she's one of the Person of the Year in Time magazine, and so that's where she started. So I think we need to have more education.
Also education of Congress.
That's impossible.
That's the next question. How do we get scientists? Actually, per capita scientists are the
If
most
you're taking,
underrepresented
delete what I said.
But we elect Congress, right? We elect these people, so you've got to make sure that, you know, it's your vote counts, and whom you're voting. What are your priorities in life? Because these people are the ones that are passing the bills and determining where the money is being put. It's not the country is not making enough money to put more money into research. It's that it's just not a priority. The budget, I mean, if we feel it every year when we get our budgets from federal fund, that it doesn't go up, it goes down, but the cost of doing science goes up. You can see us all here, we're not millionaires. And the big thing is, for some reason, there's public perception that scientists are these weirdos that don't really know what they're doing. I mean, why does people listen to celebrities about not vaccinating your kids when everyone is telling you that first paper that said the vaccination led to autism was not, it was all fabrication. So we've been spending the past 10 years trying to erase all of that, when it takes just one celebrity to say, oh no, you shouldn't vaccinate your kids, that's bad.
I use that paper, I teach a class on ethics. And I use that paper as an example of every single unethical thing you can possibly do in a paper. It wasn't just, it was fabricated. There were only 12 subjects. Most of them, it turned out later, they checked, didn't have autism. That was a diagnosis that he made.
Yeah, this was all based on recollection. If you could, do you remember when did your child first started seeing signs of autism? Because like memory is very fragile, and if I tell you that if you eat more carrots, you're going to be healthier, then your subconscious is going to say like, oh yeah, I started eating carrots and I was healthier.
I think that we should educate celebrities then. I think let's start with celebrities.
Our jobs are hard enough.
No, I think I'm gonna, that's not it, that's
One more.
So I just want to add, one thing, that the timing for the changes is perfect, because a lot of ideas that we had before were not able to implement. But right now we have lots of new technology, new tools. The patients are much more educated. For example, those watches, for example, we can measure our own sugars, heartbeat. So we go to the hospital these days. We can Google our symptoms. Symptoms are a lot already, and there's also those disease interest groups, online as well, social media. So people are really talking. So this just a lot more educated than ever before. So I think things can happen much more rapidly if you're gonna move on the right direction.
Can I have one thing to that? When you were saying about how corporations are in business to make money, I think one thing that I personally want to look into myself, and of course I'm a very, very small investor in my 401k, but it's like, we support all these corporations and what they've chosen to do by giving them our investments. And it's like, they're going to listen if they don't have any money coming in. I think that might be a tactic that worth looking at.
Yeah, I think also just writing to Congress, occasionally bills come up, you read in the paper that there's going to be a vote on a bill to increase the NIH budget by 10% or something, and usually that's
It's not that high.
Well, that's after.
What they told you.
Yeah, so the funding situation in academics is really bad these days. So in the olden days when I was more like their age, probably 30%, the odds of getting a grant from NIH to do the kind of research that we all do are probably about 1 in 3. I got a grant one time that was rated right about at the 30th percentile, and these days that would be, it's down around closer to 10% now. And so it's, despite the fact that in constant dollars the NIH budgets have increased, as somebody weighed in, he mentioned, costs have gone up too, so much that the effect of the number of grants that can be funded has really dropped like a rock in the past 20 years.
But I think the need of education can only be, I think this is the best evidence of how much education in the United States needs to change. The people that are sitting on this table right here, none of us is American. So and I appreciate that you have your own people to cover it, like the intellectual.
Well, we don't have to do it. You guys are so
but yeah, I'm sure that there's a lot of potential within your own country to advance research. It's just a matter of keeping up with education.
Well, now I have a lot of stuff. One is that writing to, we call them representatives, but they're not representatives, they're politicians. What I've found in my experience is that talking to them is useless. What you have to really do is get to the party, the Republican and Democratic Party, and get the party to support that kind of stuff. Because, especially in California, people vote the way the party tells them to. And it happens a lot in Washington too.
A lot of people just don't vote.
Well, but you have to get into the party plank and get into those things. Okay? And before I forget this part, I'm on a school board. So what should schools, K-12 schools be doing to try to help lead people into the sciences more? I know we are doing some of that, but
so I have a very special thought on this. The country, right? The government, NSF, it pushed really hard for this, STEM thing, right? Science and engineering, all this sort of thing. So I read an article in Nature. I think last year there was a person talking about this, right? So he was like, so look at the other field, like lawyers or movie stars or other things, right? You never really need to push them to say, okay, so work on this field. Why? It's because they paid so high. So so he argues that all we are doing right now are just asking the wrong question and working the wrong solution. You can just simply increase our salary. Who work in the science, engineering, math field, all of a sudden you address all these problems because you're gonna pull all the kids work on that field, right? Because so I even think it's not ethical to attract the people to work on STEM because you're fooling them. You're putting them into a horrible field. They get paid so low. They work so hard. They do their PhD, postdoc, and all of a sudden they just earn this such a poor salary. It's really bad, right?
So the one part of that is making a little bit of a comeback is in chemical engineering, electrical engineering, and computer engineering.
And petroleum engineering.
Right, and petroleum, where you can go into those sciences. But the thing is, if I'm really good at math, do I want to go make, $200,000 a year, or do I want to be a pharm-sci professor?
Right?
A part-time professor. Which I did.
So that's why I think lots of people argue that the society, the government, take advantage of the fact that we researchers are interested in doing research.
Right.
They just we know you guys like doing that, so that's why
I think
we're just treating it so badly.
Yeah, I think you might see a little bit of a change because it's predicted that the next century will be biology century, where people will really start using biology for engineering much more than they are now. The way that silicon was coming up into the year 2000, where using all that engineering.
Well, I hope so.
Yeah. So maybe there will be a little bit of a change, but this is supposed to be biology centric.
Yeah.
So.
But I want to, I understand the job market, but I want to go back to something you said in your question. You brought up K through 12. We really have one of the worst education systems in the world when it comes to elementary school, junior or high school. It's really sad that yes, when it comes to universities, maybe we offer good education, but we don't have, I have a son in high school, and I'm not going to mention this high school. I'm counting days for him to graduate from this high school because I don't think he's learning anything. This is one of the top-ranked high schools in California, and the kids who know how to memorize do really well. So what does that tell you? That we have to step back and say, what are we really teaching our kids? Because it starts there, and then they become our students, and we have to say, no, we are not memorizing, we are learning here. Critical thinking, We don't teach any of that in our high schools. We don't teach them how to think, we don't teach them how to analyze something. They become memorizers. They become lifetime memorizers.
Standardized testers.
They are trying to change that because they recognize that's what's happening. So that's what this new Common Core is supposed to help with, to make people actually think about what's going on, not just be able to regurgitate it.
Okay, so let's say they do that,
Yeah.
but these kids are still taking SAT.
Oh yeah, sure.
Okay, so we have to start maybe changing the college admissions.
Mhm.
I have students who get a very poor MCAT score.
Mhm.
They are brilliant.
Right.
They don't get into a medical school because they have poor MCAT score, and there's really no place for them. And I said, do I want to? If I were going to choose a physician, do I choose the perfect MCAT score, who I know exactly,
Mhm.
how they were,
Right.
and how he or she was as a student in my classroom, or the one with the poor MCAT score? I would choose the one with the poor MCAT score, but he or she never makes it to medical school. So yeah, we can,
Okay, I'll fix it all for you. Okay?
yes, please.
I have a regular question.
Okay, yeah.
If it's studying children, do you think that you might be able to see predictors in them that would help with people getting cancer or diseases later on in life?
Yeah, so the beauty of studying childhood cancer is that you don't have a lifetime of environmental factors that affect those cancers, right? Because there are known smokers, there are known drinkers, you haven't, they're the perfect population. And so it really gives you that cleanliness in a system where you can really tell, okay, a mutation in this gene is causing this reason. And so and all the genes that are commonly mutated in cancer, as I mentioned, the retinoblastoma susceptibility gene is just one of many, but all tend to repeat themselves. And so why, actually, the even bigger question is why some genes cause cancer in some tissues, why just retinoblastoma and not cancer anywhere else? And what's so particular? So in kids, because you don't have this life choices that affect their epigenome, might be an easier way of going about it and determine, and then jump back into adults and see what is it. And so it has been quite interesting because as a follow-up on our study that's showing that epigenetics was so key and that you only needed one mutation and then everything else happened epigenetically. Now everyone can, in studying adult cancers, have gone back and see, okay, let's forget about the genetic factors of it. Let's look at the epigenetics. And there's various different groups that are now coming and realizing that the dogmas that have been true for 20 years were not so true, and coming up with alternative therapeutics. So, I mean, this is the day and age. Technology has arrived to the point where I think we're in a great place to be doing science today. The cost of doing whole genome sequences has significantly dropped. The first time the human genome was sequenced, it cost $1 billion. Now you can sequence your genome for $10,000. It's still quite expensive, but it's nothing with respect to $4 billion.
But you can get a million SNPs from 23andMe for $99.
But it is. It's a very nice system which you can look at.
So I have a question actually for Leon. When we talk about bacteria, and so bacteria cells are 10 to 1 in humans, does it look like that the spectrum of bacteria is very broad, or are there certain types of bacteria that are very common that seem to dominate?
Yeah, well, so first of all, I'm not a microbiologist. I don't really know that question. But what I know is they're very diverse, very dynamic. It depends on person as well, and it's also depending on who you are most associated with. For example, children share lots of same bacteria with their parents, things like that. I don't have a very good answer for that question.
And also who you're kissing, right?
Your kids don't kiss, so then you even
Okay, so I want to thank you all for coming. Let's give another hand for our panel. I want to invite each of you to enjoy some spirits and some food and mingle with the faculty and each other and enjoy the rest of the evening.